Okay, so we're going to kind of dive a little bit deeper today, going into the innate and the adaptive immune system. The adaptive immune system is going to actually be covered Thursday and Friday. So Thursday we're doing B cells and Friday we're doing T cells, but it's a lot. It really is a lot. So we're going to do like a little bit of an intro today. And the innate immune system is primarily going to be today, with then a little bit more on the complement system tomorrow. I think that's what tomorrow's is. Same thing as yesterday. If you guys have any questions, stop me. Like, do not let Immuno build, because it's just not going to make sense. So stop me or just send a question in the chat, okay? And we'll stop and chat about it. Okay, so big thing to know, like right off the bat, is like your innate versus adaptive immune system. Like this is kind of like, what's an upper motor neuron lesion and what's a lower motor neuron lesion. That table you had to know. This is kind of like that like level of understanding. You kind of have to understand the difference between these two. So the innate immune system is a primitive system, and primitive just means, I think basically what it means is like everyone has it, like from cavemen, growing up to like actual humans, right? So like everyone has an innate immune system. The adaptive immune system is something that I guess adapted. It kind of like adapted with us as we were like going through evolution. So that's a big distinction right off the bat. The innate immune system responds immediately, like it's very, very quick to respond, whereas the adaptive immune system, it kind of takes a while to undergo processes of inflammation and clonal expansion, which we'll talk about. The innate immune system is not specific. So I actually think about this as like if you get an E. coli pathogen infection, or if you get a salmonella pathogen infection, or like a COVID pathogen infection, the innate immune system is going to respond the exact same way every single time. It doesn't matter which pathogen is infecting. Whereas the adaptive immune system is very, very, very specific. This is kind of like the lock and key, like with an enzyme, and it's, I don't even know, like the enzyme binding to it's whatever, they call it a lock and key. It's kind of the same concept here. It's like that specific. It's like the amino acid level of specificity. And then the innate immune system has no memory. It doesn't need memory because it responds the same way to every single pathogen. Whereas the adaptive immune system, you get memory. And this is actually one of the big things. Again, I keep bringing up vaccines, but vaccines are a really good way to understand the adaptive immune system. And that's a big goal of vaccines, is to develop memory. So when we see a pathogen again in the future, we know exactly how to kind of take care of it. So these are like the four tenets, I guess, of these two different immune systems. So innate immunity, this is kind of like a very busy chart, but I like it. It kind of has everything on here. And if you think about it from like going from top to bottom, you're going to see like different steps that are occurring. And then there's different things that need to fail to go on to the next step. So for example, the innate immune system is like the initial infection is going to occur in 3 phases, which I kind of have, it has like fail here. And a really important thing to understand with the innate immune system is that many things have to fail before we even get to the adaptive immune system. The adaptive immune system is really going to kick in like if your body needs it. But a lot of the time when we get to this inflammatory response, which is like the last, I guess, step of the innate immune system, that's usually enough to get rid of the infectious agent. So you can kind of see here with like the little tiers, like we could stop the infection at this level, and if that fails, we're going to move on. And then at this level, we could also remove it here, but if that fails, we're going to move on. And then we could also stop it here, and then if that fails, now we're going to trigger the adaptive immune response. So it's kind of like this idea of like failure before we get to the adaptive immune system. And this idea of immune exclusion, this is like you're not even allowing a pathogen to infect you. So like things like your skin is there to kind of protect against like pathogens entering. And if you have a breach in your skin, that's why they say you really should clean a cutout, take care of it, especially after surgery or something, because that's when pathogens can go in. So a lot of the times immune exclusion is good. And then if not, we have all these fail-safe mechanisms. So these are kind of like, again, this is kind of like the 3 tiers, right? So first we have our anatomic barriers. This is like that immune exclusion. So we're kind of like your skin, your mucosa, the respiratory epithelium, the intestine. I mean, bacteria and viruses can enter at various different routes. And there's always these barriers that are in place in order to try and not even let them enter. If this kind of fails, then we're going to get some antimicrobial proteins being released. Complement, we're going to talk about complement a lot tomorrow. You guys are going to be so sick of complement after tomorrow. But complement is a really good innate immune system, like thing to get rid of pathogens. If that fails, then we have some innate immune cells that are just kind of always patrolling the area. These are always kind of understanding like which cells are innate and which cells fall under the adaptive immunity, because again, these are the guys that are going to be playing first before we even get to adaptive immunity. So some innate immune cells, there's a bunch, but some examples are macrophages, dendritic cells, and natural killer cells. And I kind of put them over here for these pictures so you can kind of see what they look like. Neutrophils are also another example of an innate immune cell. And we talked about yesterday, remember, monocytes and macrophages. If it's in the blood, it's called a monocyte, and if it's in a tissue, it's called a macrophage. Dendritic cells are arguably the most important innate immune cell, and we'll talk about why, but basically these are the only cells that can kind of bridge the gap between the innate immune system and the adaptive immune system. So everyone kind of generally says that this is the most important innate immune cell because it's the only one basically that can activate the adaptive immune system. And then your natural killer cell, which we'll talk about. And then the adaptive immune system is like your B cells and your T cells, your lymphocytes, which we talked about yesterday. So innate immunity, you can think of it as occurring in a series of steps. So we talked, remember yesterday, I kind of mentioned like these terminologies that might be coming up randomly throughout the sessions. So PAMPs, remember it stands for pathogen associated molecular patterns. So these are things that only the microbes should have, and we humans like should not have them at all. So the body should be able to recognize those as being foreign. So some examples of PAMPs I put up here is LPS, which is on gram-negative bacteria. Flagellin, which is a part of bacteria, it allows them to like move around, and then some nucleic acids. Like some viruses, for example, have like double-stranded RNA, and your body's like, we don't have double-stranded RNA, that's weird, or single-stranded DNA, things like that. So they can kind of like recognize nucleic acid sequences of viruses as being foreign. So it's technically a PAMP. Even like double-stranded DNA viruses or single-stranded RNA viruses, they have certain like genetic coding, like genetic codes that the body still can recognize as being foreign. So then we talked about sentinel cells yesterday. Remember, the sentinel cells are macrophages, dendritic cells, and epithelial cells. These are kind of the first people on the scene. They're like the first responders that are sensing something's wrong. They utilize their pattern recognition receptors, so they're PRRs, and pattern recognition receptors are what actually bind to PAMPs. And some examples of pattern recognition receptors, I added this one right before because I forgot that that's also like another one that can come up sometimes. So 2 examples of pattern recognition receptors are toll-like receptors, or TLRs, and nod receptors. They're like the same thing. They just recognize different things. It doesn't matter. That specificity doesn't really matter. But these are examples of PRRs. These are what recognize the PAMPs. And the PRRs are located on your sentinel cells, and then the PAMPs are located on the like invaders. And then from here, once we get that binding, the PAMP to PRR, we're going to get a release of mediators. And again, we kind of mentioned some of these yesterday, cytokines, chemokines, and CAMs. Cytokines, these are like how cells talk to each other. Chemokines are also like how cells talk to each other, but they tell cells where to go. They're kind of like the ones that kind of initiate movement of cells. And then CAMs, these are cell adhesion molecules. So this is like if you want a cell to leave at a certain spot, you would upregulate some CAMs so that the cell can be pulled out at that exact spot. Okay, so it's like whatever you need to happen is going to happen at this step. And it's going to be because of the binding of the pattern recognition receptor to the PAMP. So TLRs are really, really, really good pattern recognition receptors. They can be located on the cell surface. I think when people think like receptor, they're thinking like, oh, here's a cell, here's its receptor. That is, TLRs can be receptor, but they can also be located like intercellularly. So they can be located in here. And these are really great PRRs because they induce this transcription factor, NF-kappa B. This is what my entire PhD research thesis is on. Good stuff. But I won't go into too much detail. You don't have to know like all of this stuff about NF-kappa B. But basically, I want you to, when you think of NF-kappa B, I want you to think inflammation. Like I want you to think like inflammatory responses occurring. NF, if anyone's wondering like what this stands for, NF, whenever you see NF, it's like nuclear factor. So you know it's going to the nucleus. It's a transcription factor. All transcription factors are going to go to the nucleus to activate transcription, right? And then this is just the family it belongs to, like the kappa whatever family. Okay, so this is like, again, NF-kappa B is involved in so many different things, but one of the main things is to produce these pro-inflammatory cytokines and try to get to that inflammatory response. And if I go back, it's because we kind of want this to happen like, I know it's like we could kind of clear the agent here, but we're going to eventually need inflammation occurring. So we kind of like are, it doesn't have to be like this exact stepwise fashion. Like if a macrophage or one of those sentinel cells recognizes it right away, it can also just kind of like activate inflammation right off the bat. And that's totally fine. It's like all of these are there to kind of act as like a fail-safe before you get the adaptive immune system. So these can happen all at the same time. They can do one versus the other. It doesn't matter. As long as you're staying above this line, that's like kind of what their role is. Okay. So for NF-kappa B, one of the things that it does is it produces an inflammatory response. And we'll talk about inflammation in a little bit. But I wanted to kind of show you how like TLRs work with like PAMPs. So here, don't memorize this pathway. I'm putting this out right now. Do not memorize this. This is something that like last year's block director kind of wanted me to go into more detail on, but I left it because it's at least good to kind of visualize it. So here's like a macrophage. And we talked about the sentinel cells, including macrophages, have these toll-like receptors on their surface or intracellularly. This is a must know. You must know that TLR4, which is a toll-like receptor, it's a PRR. Another name for it is CD14, and it recognizes LPS. That's just like the classic one. Like you'll get boards questions on that. Like that's just like the classic one they expect you to know. So TLR4 and CD14, you can say these interchangeably. And they recognize specifically LPS. Like that's the PAMP that it recognizes. So here's LPS. It's just on the surface of some bacteria. It's going to bind to the toll-like receptor. This gene, and I only put it here because there's a pathology state where if you have this gene's like knocked out, this protein's knocked out, you get the same response. But basically this protein gets activated, and within the macrophage, we have this whole cascade of events that happen. Don't memorize it. But at the end of it, you're going to release NF-kappa B. Again, releasing NF-kappa B, it's a transcription factor. It's going to go to the nucleus, and it's going to bind to DNA, and it's going to upregulate the transcription of pro-inflammatory cytokines, which we're going to talk about today, like what pro-inflammatory cytokines are. If anything goes wrong with this pathway, there's a condition called NEMO. I know it's a really scary looking picture, but NEMO deficiency it stands for NF-kappa B essential modulator deficiency. So this is saying an essential modulator of the NF-kappa B pathway can be deficient. So if I go back here, like I have IRAK4 here, I have IKK here, like again, I don't think you need to know the specifics, but one of these are deficient, dysfunctional, so you're getting this impaired NF-kappa B response. And for whatever reason, that presents with a syndrome called ectodermal dysplasia. So this is where the patient has very thick and dry skin. They have these conical looking teeth. They have no sweat glands, so that you'll see this is like hypohidrosis. And then they just have like a wide range of infections. And this is the whole idea, like you can't stimulate like an inflammatory response. So they're gonna constantly be infected with stuff. And then so this is kind of like IRAK4 is out, IKK is out, and then just this whole pathway doesn't work. Okay, so that's kind of like a pathology state associated with the NF-kappa B pathway. But again, NF-kappa B, think inflammation, pro-inflammatory cytokines. All right, so here are some more need to knows. So these are some cells in the innate immune system that are important. And it's really important to know these roles because when you're doing questions or whatever, you can kind of figure out what cell they're talking about or what cell would be involved by understanding the roles. So I kind of broke it up into 3 main roles of these cells, and then we can kind of group them based on this. So there's phagocytic cells, and phagocytic, I kind of mentioned it yesterday at the end with the reticuloendothelial system, but phago means eat. So these are cells that literally engulf pathogens. Like they have the ability to kind of change their cytoplasm to like engulf and literally eat pathogens. The main phagocytic cell, and I put 3 stars next to it, is neutrophils. They're very good at phagocytosis. They're very, very good at it. But macrophages can also phagocytose. Dendritic cells can also phagocytose. And eosinophils technically can, and it's in this thing that they can, but like no one thinks about them as being phagocytic cells. So you're good to just kind of think of these 3. But for completeness, I put eosinophils. Eosinophils have another role. Okay, so those are the phagocytic cells that can go and engulf pathogens. Next, we have antigen-presenting cells. These are need-to-knows, and I will ask you this multiple times throughout the entire rest of the sessions. You need to know the 3 APCs. So antigen-presenting cells, these are the cells that present antigen to either a T cell or a B cell. So remember I talked about dendritic cell. This is the absolute most important one. This is why it's like its main function is to do this. Macrophages can also do this. We kind of talked about this yesterday. Like it can come in through those high endothelial venules, right, and in the lymph nodes, and it can go present antigen to a T cell or a B cell. And then, confusingly, which is great, but it's like going to take a while to kind of like just remember that B cells can technically also be antigen presenting cells, which is super annoying because they're not innate immune cells, they're adaptive immune cells, but they can be. And they can present antigen to, obviously they can't present antigen to themselves, but they can present antigen to T cells. But we'll talk about that with B cells on Thursday, but just these are 3 you definitely need to know: macrophages, dendritic cells, and B cells. Sometimes you might even just get a question like, what are the 3 antigen present? Like literally, like that's how important it is to know it. And then finally, you have mediator releasing cells. So these are cells that release mediators, which I know, but basically like you can think of it like eosinophils. The mediator that they release is, well, we'll talk about it, but it's like it's involved in like parasitic infection. So the mediator they release is going to help with parasitic infections. And again, we'll talk about that. Basophils, these are going to release things that are involved in like the allergic responses and also in the anti-parasitic, in the parasitic responses. And then mast cells, these are the last guys. Mast cells are also involved in mediator releasing, and I hope you guys all know that mast cells are what's responsible for releasing histamine. So if you've ever had allergies, this is what's making you have the symptoms of allergies. And we'll talk about histamine today a little bit. But that's what's meant by like mediator releasing. It's going to release something. Like that's its main function, is to release something to go do something. In easy terms to explain it.
Christina, I have a question.
Yeah.
Like the difference between a basophil and a mast cell. So like I notice sometimes like when you're looking at a lineage picture, it will
Mhm.
only say basophil because then they're saying it's also a mast cell. So like what's like the difference and the similarities?
Yeah, that's actually, I meant to point that out, so thank you. Mast cells are only in the tissues. So there's also a thing, like there's also something that might come up with like if you have asthma, you might have an elevation in eosinophils, you might have an elevation in basophils, and you might have an increase in mast cells. And sometimes they'll get to by asking like the blood, like what's going to be elevated in the blood. Mast cells are not in the blood, they're only in the tissues. So that's like pretty much the main distinction. Is it kind of like, oh, sorry, is it kind of like the monocyte to macrophage thing? Kind of, yeah. Basophil, yeah, because as Naomi said, they're kind of like the lineage. It's kind of like they do one or the other. You can think of it exactly like that. It's just one's in the tissues and one's in the blood. So yeah, kind of like the monocyte macrophage thing, except I don't think like a basophil itself can turn into a mast cell. Whereas like with the monocyte macrophage thing, a monocyte itself turns into a macrophage, if that makes sense. But basophils and mast cells do come from the same lineage for sure. And then something not shown here, NK cells, these are really, really important innate immune cells. I have like a slide or 2 dedicated to them. They're really important. They're kind of like the T cells of the innate immune system. They're a really important cell, but I guess it's not important to know what they look like, I guess, but we'll talk about them as well. Okay, but the phagocytic cells, macrophages, dendritic cells, neutrophils, APCs, macrophages, dendritic cell, B cells, and your mediator releasing cells, your eosinophils, basophils, mast cells. Okay. Okay. So for these 3 things, kind of going into a little bit more. So phagocytosis, again, macrophages, dendritic cell, neutrophils, eosinophils, sure, but again, the main 3, macrophages, dendritic cells, neutrophils. Steps of phagocytosis, and I didn't go into too much detail whatsoever. I just kind of wanted you to like understand what it is. So here's like, they're saying this is entrapment of a food particle. That's fine. If it was a bacteria, like a bacteria would be the same process. So here's a food particle / bacteria. The macrophage and dendritic cell or neutrophil is going to literally change its cytoplasm to eat it, literally grab it, and then it's going to bring it into a vesicle. It's called a phagosome. So now it's in a phagosome. And then there's these lysosomes, and lysosomes contain digestive enzymes. The lysosomes are going to fuse with the phagosome, and then you're going to get a phagolysosome, and that's going to digest whatever it is. It's going to digest the food particle, it's going to digest the bacteria, and so forth. But that's the general process. You go from phagosome to phagolysosome to complete just destruction. Antigen presenting cells. So this is like kind of how the process of like antigen presentation works if you're an antigen presenting cell. So like I kind of said here, macrophages and dendritic cells are phagocytic and they're antigen presenting. So what this means is like the macrophage or the dendritic cell can engulf the bacteria, go through this process, and then when it digests it at this point, it's going to take some of those broken down peptides and it's not going to destroy them all. I mean, you want destruction. You do want to get rid of that pathogen, but you also want to let the adaptive immune system like be like, hey guys, like this just tried to infect us. So if you ever see this again, I'm going to need you to like shut that down immediately. So what it does is it's going to load it onto what's known as an MHC class 2 molecule. And I'm just throwing out terms, but we will go into more detail. So from here, there's good like this whole loading process onto MHC class 2, and you do need to know it's class 2. There's also a class 1 that's important. So it's going to load it onto like an MHC class 2, like all on its surface, and then it's going to go basically find a T cell. So it's going to go travel to the secondary lymphoid tissue and present that to a T cell. So that's that whole idea of antigen presentation. And again, remember an antigen is anything like foreign, but it's not an immune. Remember, antigens are like subsets of immunogens, so it's going to get an immune response. It's something that the body recognizes as being foreign. And then it presents it to a T cell. We will go into more detail on Friday. And then the mediator releases. The releasers are your mast cells, basophils, eosinophils. These are going to release mediators. And again, it's like a lot of these do play a role in allergic reactions, but like eosinophils, basophils, they can play a role in parasitic infections, right? So they can release specific mediators for like that are anti-parasitic, which we'll talk about in a little bit. This is just like a general overview. Okay. So again, here's kind of like the big point behind dendritic cells, why they're like the most important cell ever, is because they bridge that gap between the innate and adaptive immune system. And that's kind of what this is saying here. They form the bridge between these two responses. So there's different types of cells. There's like these granulocytes, the monocytes, or the macrophages, right? Then dendritic cell is really good. So the reason why, because I mean, we were talking about how like B cells and macrophages can also present antigen to T cells. The reason why dendritic cells are known as like the best, the one that can do this is like the entire role of the antigen of the dendritic cell. This is what the dendritic cell lives for. It lives to present antigen. So sometimes they call it like a dedicated antigen presenting cell because this is the only thing it does. I mean, we talked about macrophages, how they can be involved with like some other stuff, like with the red blood cells from yesterday. I remember with the spleen. So they can be involved in some other stuff. Dendritic cells, this is all they do all day. So they're like professional APCs. So that's why it's like they're like the most important one. They're like the best one at doing this, and they bridge that gap between these two systems. So again, it's kind of like that whole idea, like with this, they're gonna load it and like they're still gonna take care of the pathogen. They can still get rid of it, but they're still gonna go tell like the T cells and B cells like, hey, this just infected us. Can you just kind of make some memory against it for next time? And again, that's adaptive immunity. We'll touch on it a little bit today, but more so on Thursday and Friday with B cells and T cells. Okay. So some important innate immune cytokines. This is kind of where Immuno loses some of its audience. You know, you have to memorize you really have to memorize so much, and it's really obnoxious. Flashcards, whatever works for you, but you do kind of need to know some of these. They do become clinically relevant. Like you'll learn with some drugs that inhibit certain cytokines, like even with COVID right now, they're finding that there's a certain cytokine that's driving like this detrimental immune responses. So people developed a drug against that one cytokine. So it is kind of good to know these, but I do know there's a lot. So I tried to come up with some that are at least like you should 100% know these for your exam or for, you know, just for life. So these important immune cytokines, you can kind of think of this is my really poorly drawn macrophage. I just didn't want to have a blank screen. So these macrophages are going to secrete so these macrophages are going to secrete, they're going to secrete acute, like I say acute then recruit. So they release these like cytokines to do something acutely, and usually it's some sort of inflammation, which you're gonna see in a second. Like these all involved with inflammation. That's what's meant by acute. They want to do something to kind of take care of the action right now. So they're like, okay, let's initiate an inflammatory response. So that's IL-1, IL-6, and TNF alpha. Those are like the main three. Then they're going to recruit other cells to the area. So like, let's say the macrophage came upon like a virus. You're going to want like certain cells coming to that area to take care of a virus versus if the macrophage came into contact with a worm versus a bacteria. So that's this whole idea of like recruiting the proper cell. So the main ones here are IL-8 or CXCL8. They're the same thing. It's really annoying. IL-8 is like a cytokine. CXCL8 is like the chemokine version of it. It's not a big deal. They both have 8 in the name, so it's fine. But they're like the exact same thing. And then IL-12. These are like going to recruit certain cells to come to the area. So here's like more in depth. So again, acute. So IL-1 is going to cause fever and inflammation. Again, you're going to, like I kind of mentioned, that's the whole point of these acute guys, is to induce some sort of inflammatory response. But then there's like little other things with these certain cytokines, these interleukins. So IL-1 also is going to activate endothelial cells to upregulate CAMs. So remember, with CAMs, that's important because if we want to recruit in like neutrophils, for example, we need them to come out the right area. So they need to be told where to like leave the blood and come out to the tissue. And that's by CAMs being upregulated, those cell adhesion molecules. So like here's well, it's not very good. Here's like an endothelial cell in the blood vessel, right? And we're going to talk about this like in a little bit. But here's like the blood vessel, and it's lined by endothelial cells. They're going to upregulate these CAMs on its surface. So when the neutrophils like coming in like 1,000 miles an hour, it's going to basically get like speed bumped by these, slow down, and then it can kind of get pulled out to like where the infection is happening. That's the idea of CAMs. So that's why it's really important like that IL-1 does get activated like immediately from these macrophages. And then they do other stuff. They can get other chemokines to be secreted. IL-6 causes production of acute phase reactants. I talk about those in a little bit, what those are. And then TNF alpha is going to basically, the main thing with TNF alpha is inflammation. It has some roles in TB. And actually, like if someone has TB, they are very cachectic. You might have even seen this in like a vignette, is like cachexia. And this is just kind of, you can think of cachexia as like wasting away. Like the person just looks like they're literally like wasting away. That's like 100% due to TNF alpha. It's just, it causes cachexia. I'm not entirely sure how it does that, but it's the cytokine responsible for cachexia. And it maintains granulomas in TB. We'll talk about granulomas a little bit more on Friday with T cells, because T cells are really important with granulomas. Okay, and then these other three, IL-8 or CXCL8, this is neutrophils. So the way to kind of remember IL-8 is clean up on IL-8. So IL, it's clever. And clean up because neutrophils are the ones that kind of gobble up things. So they can gobble up the trash in that area. So clean up on IL-8 to call in your neutrophils. IL-12 is going to activate your NK cells. Don't worry about IL-15. I think this is something on like the study guide from last year, but don't worry about it. IL-12 is the more important one. This is going to recruit NK cells to the area. Okay. And then we'll talk about IL-12 a little bit more with T cell differentiation and activation. These are like the main things that it does. Like as soon as the macrophage gets activated and releases these cytokines, this is what it's secreting. So this is kind of what I just said, but in a picture would have been a lot easier. But this is, at the end of the day, what we're doing is we are getting inflammation at the site of the infection. And again, remember, a lot of the times this is enough to get rid of the pathogen. So here is just a little bit more on like IL-12 and CXCL8. So here's your macrophage, it ingested a virus. And again, we'll talk about NK cells, but NK cells are like the T cells of the innate immune system. So they're really good at clearing out viruses, which we'll talk about in a little bit. So this macrophage is going to secrete IL-12 and CXCL8. NK cell is going to come into contact with it. It's going to release more IL-12. They're just going to kind of go back and forth. And then you're going to get eventual destruction of the pathogens within it. Interferon gamma is, you might see this coming up a little bit. I think we're going to talk about it more on Friday again with granulomas. But interferon gamma is, you can think of this as making macrophages really good killers. That's like the best way to think of what interferon gamma is. Not only NK cells release interferon gamma, like a lot of cells can release interferon gamma, but whenever it's released and there's a macrophage in the area, the whole purpose of interferon gamma is to make the macrophage good at killing. So for example, here is like the virus just in like a phagosome, right? So the macrophage ingested it. Maybe the virus is hiding from the lysosomes, or maybe it's not able to kill the virus. It's just too good of a virus. So like that digestion step isn't going to happen. So what can happen is a nearby cell can secrete interferon gamma and actually make this macrophage really good at killing. And then it can actually digest the virus and kill some of it, which is what it's kind of showing here. Okay, so this is just an example of how like these cytokines are like involved. Here is this is the first aid cytokines that first aid says is important. And these are definitely the ones that you should know for boards. I just talked to someone that took boards like 2 weeks ago and said like, these are the ones that they asked about. So these are good ones to know. So the mnemonic is hot T-bone steak. So I put them in order and like what it's like talking about, but hot T-bone steak. So you basically go in order from IL-1 to IL-6. IL-1 induces a fever. And again, these cytokines do other things, but it's like this is like the main thing it's associated with. So like I said, IL-1 also upregulates CAMs, right? So it's kind of like this is just for first aid's way of helping you remember it. But IL-1 induces a fever, so that's hot. IL-2 is called T cell growth factor. This one is actually like if you want more T cells in the area, you can actually release IL-2 and like T cells will generate even more from like out from the bone marrow. So they call it T cell growth factor. IL-3 stimulates bone marrow, so this is hot T-bone, and then steak. So IL-4 stimulates IgE, IL-5 IgA. I think we talked about this on Thursday. If you're like, what is IgA and IgA? We'll talk about this on Thursday with antibodies. And then IL-6 stimulates acute phase reactants. It's not spelled like this, so don't spell it like this, but it's just a nice way to remember it. And acute phase reactants, we'll talk about those, I think, in a little bit. Those are kind of released anytime there's inflammation. The liver just upregulates acute phase reactants. And the whole idea, again, you're just trying to get more inflammation going, basically. Okay, so that's IL-1 through IL-6 and kind of like just like one function that it's associated with. Lots of stuff. All right. So next we're going to talk about inflammation. You should know the inflammatory response. I'm going to just kind of go into it in a little bit more detail. So inflammation, nonspecific, we know that it's a part of the innate immune system, right? So it's nonspecific response to injury or an invasion. I'm sure any of you guys have either, gotten bit by a mosquito or rolled your ankle. Like, you know what happens when you have inflammation. So like the other things that happen during inflammation are you have migrations of neutrophils to the area. You can even get fibrin clots. So like that's more hemonc, but like you can get like these clots happening. Like let's say you cut your skin, and the inflammatory response can actually stimulate like that process of coagulation, which again, that's next year. But this is like that's another thing that the inflammatory response can do. Macrophages, obviously huge players in this. And then the process of like healing is also involved with the inflammatory response. There are 5 cardinal signs of inflammation. You should know these. I don't really know if it's like they're still asking the Latin versions of these. I'm not really sure, but I put them here just because sometimes you see the Latin versions of these. I don't know. But so it's basically redness or rubor, heat, calor, swelling, tumor, and pain, which is dolor. Those are like the ors, right? And then the last one is like loss of function. So you can't use that whatever it is. Like let's say you rolled your ankle. You can't use your ankle for the time being. You have to kind of let it go through the process. But you don't really have to remember these. Just kind of think about like anytime you've ever like hurt yourself or like had like some sort of like you touched your skin, it was really hot because you injured yourself there. That's kind of like inflammation happening. But to kind of visualize it, I'm a visual person, right? So we have the barrier. We have our skin. This is a barrier, like the first line of defense, right, against any microbe. This is the first part of the innate immune response. So the skin is keeping out the microbes. So here are the microbes in green. It's keeping them out. It's looking great. But let's say you prick yourself with a thorn. Prick yourself with a thorn. Oh, now I just basically allowed a pathway for the microbes on my skin to get in. So now they've breached that barrier. So step 1 failed. They failed the immune exclusion thing, right? So now we kind of need step 2 and 3 to kind of kick in. We need the innate immune cells to kind of get activated. Maybe we need some complement happening, like depending on the situation. Just something else needs to happen at this point to kind of try and get rid of these microbes. So one of the first things that happens is mast cells, these are tissue, right? So these are located in the tissue. They're right here. They're actually going to secrete histamine. So these guys are going to secrete histamine, which is going to go to the blood, and it's going to cause vasodilation. This is kind of what's responsible for the redness and the heat aspect of the inflammatory response. Also, it's not on here. I just didn't have enough time to put it in here, but macrophages can also secrete we talked about this, IL-1, like TNF alpha, IL-6, it can also secrete some stuff, but the end result is the same. You get this vasodilation of the blood vessels that are located right there. The second thing that happens is that these are going to get really leaky. So the endothelium, so this is in red, so I'm not going to draw it on it, But blood vessels are lined with endothelial cells. So they're lined like this, and they're really, really closely, tightly packed together. It's not that tall. Okay. But so when you have like certain cytokines being released, when histamine gets released and these get bigger, the endothelial cells actually get like spread apart a lot more. And this is what kind of leads to the swelling. This is kind of what leads to the swelling of, the inflammatory response. Oh, you had this question today on a practice boards test? It's very cool. Yeah, someone just chatted me that she had this question today. Yeah, so they do like to ask about inflammatory response. I'm telling you, they love Immuno. So anyway, this is like the whole leakiness aspect, right? And why do we need the endothelial cells to be leakier? Because we need certain things to leave the blood to get to the site. So what's leaking are these endothelial cells. So the blood vessel, so this blood vessel, I just didn't draw it on this figure. Blood vessels are lined with endothelial cells. So they're lined like this. And when these mediators are released, so histamine, IL-1, TNF alpha, all that stuff, when they're released and go to the blood, they cause vasodilation of the blood, and they also cause the endothelial cells to kind of like spread out. So when they spread out, now this blood is like leaky. Now it's allowing for things to kind of leave. We don't have that tight junctional barrier anymore between those endothelial cells. So now like things are like free to leave the blood. So that's kind of what's meant by like leakiness, and that's what's responsible for the swelling, cardinal sign of inflammation. Okay. So one of the things that can leave now that it has room to leave is platelets and clotting factors. Don't worry too much about this. Remember, you're going to do this next year. But let's say you actually cut yourself. They need to plug the clot. So that's what platelets are going to do. And then let's say, so the macrophage secreted the acute cytokines, right? Now it's going to secrete the recruit cytokines. So something like CXCL8 or IL-8, right? This is your cleanup on IL-8. This is your neutrophil chemotactic factor. So here's your neutrophil. Here it comes. It's like, okay, and it's going to leave at that site of, it's going to leave at the site of the infection because one of these other acute cytokines released was IL-1. And remember, IL-1 upregulates the CAMs on the surface of these endothelial cells, the cell adhesion molecules. So the neutrophil is going to go, it's going to basically slow down because the CAMs are going to act like speed bumps, and then it's going to like leave at that exact site. And then once it's here, these are like the garbage disposal guys, right? They're going to go up, they're going to like phagocytose these bacteria, and they're going to basically, this is what's kind of leading to that pain and loss of function because like the neutrophils are kind of doing its job. It's trying to eat up all the microbes within this area. Neutrophils too, like if you've ever had like pus form from like any infection you've ever had, like an abscess or something, that's neutrophils. All like pus is like dead neutrophils because it came to the area, it gobbled up whatever was there, and then they die when they're done, basically. They basically die, and then actually macrophages like eat them up, but we'll talk about that. But yeah, so that's that. This is the same thing, but a little bit harder to kind of see because it's just a lot of things happening on here. But this whole process, so like this is kind of what I meant. Here's your blood vessel. Here's like right here. And then it's not showing it, but there would be like the top part up here. And then you have these endothelial cells here. And that's what gets leaky when you have like the release of those mediators. So when the neutrophil comes into the area, it's not like super quick like this. It's not like really, really, really quick. It goes through this series of steps. It goes through this series of steps. So the series of steps are margination, rolling, adhesion, and diapedesis. So this is talking about the neutrophil going from the blood and then eventually leaving at the site of infection. So margination is just, again, we have like a giant blood vessel and the neutrophil is coming through. It doesn't really know where it's going. It's just kind of coming through. So it's like just like anywhere. And then when it gets to like the site of where like the infection is, it starts to get a little bit closer to like where it should be leaving. It just gets a little closer. That's called margination. So I remember this with like it's getting closer to the margin, right? It's getting closer to like that margin of endothelial cells. So that's your margination. Then we have rolling. And remember I talked about these speed bumps that's happening. So they can be CAMs, they can roll on CAMs, but there's also, it's called like they're called selectins. So it's like they're like the exact same idea as what CAMs are. They just kind of slow down the neutrophil. We're going to talk about an immunodeficiency, actually, where neutrophils lack a selectin. So selectins, if they lack a selectin, you can imagine neutrophils can't leave the area, and you can imagine these patients are going to have issues with like recurrent infections over and over. So that's why I mentioned the selectins, because this is like, it's going to come up later with with an immunodeficiency. Okay. So these selectins, they're coming, they're going to like start rolling as they go through. And then eventually, yeah, so here's a selectin here, and it's just going to kind of like, it doesn't really bind. This is just kind of showing it binding, but I think it's trying to show it in slow motion. It's just kind of slowing it down. It's like knocking into like things on the surface of the neutrophil, and it slows it down. The cams, this is what's like this is like literally grabbing the neutrophil. It's like grabbing it and pulling it in, and it binds to different types of cell surface markers. I don't think you have to know, I don't think there's an immunodeficiency that you have to know the integrin, but like it binds certain things on the neutrophil to pull it in. And then through this, the last phase of this is called diapedesis or extravasation. You might hear either one of those terms, diapedesis, extravasation. This is when the neutrophil is leaving the blood, so it's leaving out here, and it's actually going through. This process right here of it like squeezing through the endothelial cells, this is called diapedesis. Okay, so it's like literally squeezing through, and now it's at the site of injury. So it followed these chemokines, right? So it followed the gradient. Now it's here, so there must be like a microbe there for it to ingest. Okay, so this is basically just for neutrophils. This is like the pathway that a neutrophil takes to get to the site of infection.
Christina, quick
Yeah.
question.
Yeah.
For margination, would you say like IL-8 is a key player in that, or would it be
Oh,
like,
yeah, I would think so. Yeah, I think it can kind of sense that it's getting close to where it needs to be, because it will start to get a little bit closer. But yeah, IL-8 should bind to like neutrophils have receptors for IL-8. So you're not getting that tight binding at the margination point, but you can see it's probably still trying to follow this path, right? So it's like basically still trying to follow a path, and then it'll get to the site at the end. Yeah, at the end of it. Sorry. Can you explain why this causes swelling? Does it have something to do with messing up NAK pumps? That's a good question. No, it has nothing to do with the NAK pumps. So that's a really good question. When you mess with NAK pumps, this is actually something that's going to come up a lot with like your next block in Hem/Onc. As soon as you get rid of NAK pumps in a cell, like water's going to rush in and it's going to get swollen. This is different. The swelling aspect is strictly from the leakiness of these endothelial cells kind of just pulling apart a little bit. So again, your endothelial cells super, super, super tight. This is like what's lining the blood vessels. And after like the, IL-1, IL-6, TNF alpha, after all of these like pro-inflammatory cytokines are released, the endothelial cells kind of like break away from each other. So kind of like this, like it's making like these little holes now in the blood, not actual holes, but it's making like little exit points within the blood vessel. And this is leading to leakiness because now things can like basically leak out of here and go into the tissue. And then once it's in the tissue, that can present as swelling. So that's kind of what's meant by leakiness. That's a good question though. Yeah, because that is important. That's going to be important next year with the NAK pumps. But no, this is just strictly like histamine is telling the blood that it needs to get more dilated. And then by getting more dilated, it's pulling apart those endothelial cells. And it's making these little gaps. Okay, good. Okay. Yeah, no problem. So acute phase reactants, so you can see these is called APRs. I had these on like one of the other slides with like what IL-6 does. So you should associate IL-6 with acute phase reactants. So this is the K in steak, hot T-bone steak. This is the K for acute, but we can call it acute if we want to remember hot T-bone steak. So these are, again, just inflammatory markers. Like they're actually better for like testing. Like it's actually better to like, if you're in the hospital and you want to see if someone's having an inflammatory response, you would actually just order a test for one of these acute phase reactants. It's like a tangible way to see if they're having if they're having like an inflammatory response. Because like you'd be like, oh, well, can't we just do a test for cytokines? You really can't test for cytokines. It's kind of weird. You just can't test for them. You can, but they're like very expensive. So what you can do instead is you can just order a test for an acute phase reactant. I'm sure I don't know if you guys have done this yet, but you will do this a lot in Hem/Onc, is order a C-reactive protein or a CRP. That's an acute phase reactant. And the reason you order this test, the reason you order CRP is because you want to see if there's inflammation happening. Okay, so acute phase reactants, these are mostly due to IL-6, but of course the other guys can kind of play a role with it. They're all kind of interchangeable and they all have like little like unique functions. They're either going to be increased or decreased in the serum during times of acute inflammation. So this aspect of acute phase reactants being decreased during inflammation sounds like weird, but if whatever your acute phase reactant is, if there's an inflammatory response, let's say it gets gobbled up by the inflammatory response. Let's say it's like a platelet or something, right? Platelets aren't acute phase reactants. But let's say like there's something that would be involved with something in inflammation. It would actually go down, right? It would go down because it's going to the inflammatory process. But mostly acute phase reactants are increased. And that's like why you can test for it. What does CRP tell you that white cell count doesn't? Yeah, no, it's a good question. So white cells can be elevated for like different reasons. Like you can even have just like cancers that elevate your white count. White counts also just kind of tell you all of the cells. So it's telling you like neutrophils, lymphocytes, basophils, eosinophils, right? CRP is telling you like there is a specific inflammatory process occurring. So there's still inflammation of some way, shape, or form happening, which I guess the neutrophil count could tell you that too, because that's basically what neutrophils are, is like it's going to the area causing inflammation. So I guess it's just like another way. It really doesn't tell you much different from like the white count, but just keep in mind like white counts can fluctuate. White counts can even fluctuate if the patient's on a certain like medication, which I think in the last lecture we talked about. Steroids actually really, really, really increase your white count. So how do I know if it's actually like I'm on a steroid? Is that why my white count's up? Or is it because like, I'm on this medication? Or is it because like this patient has inflammation? So it's just kind of like nothing really affects CRP, C-reactive protein. So that's why it's like another good kind of like marker to see if inflammation's occurring. But yeah, it's a good question. It's a good economical question. Just, why are we ordering all these tests? But yeah, so that's why you would order a CRP. It's just kind of like, I guess like another way to see if there's inflammation occurring in the body. Yeah, no problem. CRP actually acts as an opsonin, and we're going to talk a lot about opsonins tomorrow with complement. Like you're going to love opsonins, but CRP can act as an opsonin and can kind of goes along with the complement system. So here's what we talked about: IL-1, IL-6, TNF-alpha. Remember, they're all kind of involved in these things, but remember, IL-6 primarily is for the acute phase reactants, and then eventually we'll get to complement and opsonization tomorrow. And then IL-1 and TNF-alpha and IL-6, honestly, they all kind of help with production of a fever. So that's what basically they're trying to do. They're trying to like make your body an inhabitable environment for the virus or the bacteria, so it like leaves or it just doesn't function as great. So here's kind of summary of inflammation. So bacteria are going to trigger macrophages. So macrophages are going to release the acute, then recruit cytokines and chemokines. Also, you get the mast cell degranulation. Here's a good picture of like the leakiness, right? So here's like your endothelial cells lining your blood vessel. And then once these cytokines and chemokines are released, see how like they kind of like break apart just a little bit. So now things can kind of like leak out into the tissue area. And that's kind of what causes like the swelling that you see with the inflammatory response. So vasodilation happens, we get redness, we get heat, we get swelling, we get all sorts of things. Neutrophils are going to start coming to the area. Remember, they go through margination, rolling, tight adhesion, and then diapedesis. They go through that whole process. We get the upregulation of CAMs. I kind of threw that in here too, because again, we need the CAMs to kind of grab the neutrophils to pull them out. And then eventually the whatever cells you just kind of call to the area are going to go to that area and then release whatever mediator it needs to release to kind of help get rid of that pathogen. But in the meantime, you're going to get pain, you're going to get like loss of function. So we just covered all of those 5 cardinal signs of inflammation. Okay. Okay. Make sense? Innate immunity. We love it. Okay. So this is what we just basically talked about. So we talked about the anatomic barriers. This is like your immune exclusion. We didn't talk about complement. We'll talk about it tomorrow, but just other parts of the innate immune system. And then all these innate immune cells, including like, activation of inflammation and all sorts of stuff. All of these things basically have to fail to get this adaptive immune response, to get B cells and T cells to kind of come to the area and like take care of whatever's going on. So that's what we're going to talk about now, the adaptive immunity. And again, we will go into so much more detail, I promise, with B cells and T cells. B cell activation and T cell activation is pretty important. So they kind of get their own like little lectures. Okay. So B cells, these are kind of like the snapshots of what like the difference between B cells and T cells. So both of these are lymphocytes, right? We talked about lymphocytes yesterday. They're both types of lymphocytes, and if you looked on a blood smear, you can't tell the difference between a B, like you'll be like, oh yeah, that's a lymphocyte, but you can't tell just by looking at it if it's a B cell or a T cell. They look exactly the same on blood smears. B cells are involved with what's known as the humoral immune response or humoral immunity. And humoral, it's basically just talking about like the humors of the body, like the extracellular environment. That's what humors means. So B cells are classically known to be involved with the humoral immune response. B cells are also APCs, so don't forget they're also antigen presenting cells, so they can recognize and present antigen over to T cells. And then B cells also have effector roles. So the effector roles meaning like if you activate a B cell, it can now do two things. It can either turn into a plasma cell, which is just basically an antibody secreting factory where it just churns out just all day, it's just churning out antibodies. And you can also get production of a memory B cell. And again, a lot of the times with the adaptive immune response, you're going to get memory. So memory B cell, it's going to accelerate future response to antigens, meaning like if you see that antigen again in the future, the B cell is going to remember it if you go through this like response. Okay, so that's B cells. T cells are involved in the cellular immune response. So they're involved with cellular immunity. I kind of briefly mentioned this, but they're really good at like killing things that are intracellular. So like viruses, parasites, anything that's like inside of a cell, the T cell kind of needs to kill. There's two main with quotes, main types of T cells. There's a lot of different types of T cells we're going to talk about on Friday, and you do need to know all the different types, but the main types that you'll probably see the most often are CD4 positive and CD8 positive T cells. So CD4 positive T cells, these are also known as helper T cells. So you could kind of see that interchangeably. I don't even see anymore like helper T cell like written out. I always see CD4 positive T cell, but that's the type of T cell it's referencing. It's a helper T cell. What do helper T cells do? They help. So yeah, so they help B cells make antibodies. So they basically make B cells better. Like they help B cells be better B cells. They can also produce cytokines to kind of recruit other cells to the area. All these cells can like secrete cytokines, but that's like what helper T cells are. They're kind of just like helping out. The CD8 positive T cells, these are your cytotoxic T cells, again, interchangeable. So you can see either CD8 positive or cytotoxic, same thing. These guys are the guys actually killing. So they're going to the target cells. They're going to go to like whatever cell is infected with a virus. They're going to find it and they're going to kill it. And we'll talk about that process. We'll talk about like what porphyrin and granzyme are, but it's just how it basically kills. Okay. So B cells, T cells, they're both lymphocytes, but they play these very like different roles in the adaptive immune system. So in terms of the receptors, so there's two, obviously the B cell has its own receptor and then the T cell has its own receptor. So the B cell receptor is basically just an antibody. Like you can even like put like antibody, like it's an antibody. It's just on the surface of the B cell. And then you can secrete it, and that's, you know, the antibodies that we all know and love. But when the B cell's made and when it has its receptor, it's basically just an antibody on the receptor of the B cell. So here's your B cell, here's your B cell, and then here would be like your B cell receptor, which is just think of like an antibody like tethered to a B cell. So we're going to talk about like these different, like the different parts of an antibody. We'll go into some detail of it, but I just kind of wanted you to see like, just kind of see the B cell receptor versus the T cell receptor and see how they kind of differ. But you can kind of see, like, I like this picture because it color codes it, and you can kind of see like it is kind of similar, but it's different. So for example, the B cell receptor has two arms, and both arms, these are like, they're called FAB regions, and then it has one FC region. Again, we'll talk about that, I think, on the next slide. And then the T cell receptor, it's not FAB and FC, it's just alpha. There's an alpha chain and there's a beta chain. So going into a little bit more detail of like what the B cell receptor is, this is an antibody. Another name for an antibody is an immunoglobulin, so like IG. So that's why a lot of the antibody names start with IG something. It just stands for immunoglobulin or antigen. So there's 4 chains. There's 2 heavy chains, 2 light chains. So the heavy chains are in green in this picture, and then the light chains are in yellow. I tried to color code it. And then that FAB and FC, what it stands for. So FAB stands for fragment of antigen binding, and FC stands for fragment crystallizable. I know it's not as, it doesn't tell you what it does as well as the FAB. But basically the FAB, this is what's binding to antigen. Like this is what is going to recognize like the antigen and then bind to it. Versus the FC portion down here, this, like at least with the receptor, this is what's bound to the B cell. Okay, so if you were to draw it on top of the B cell, this would be the FC portion here, and then the FAB portions would be at the top. And then I think yesterday I mentioned like epitope paratope thing. Like I kind of mentioned that terminology. The paratope is going to be on the FAB. So it's basically the part of the FAB, like the part of the FAB, part of the receptor, that's going to recognize the epitope. So it's like the exact portion on the FAB that recognizes the epitope. So they say like the paratope or this FAB domain, this is what's conferring specificity of the B cell receptor. The FC portion doesn't tell you like what it's it doesn't change. Like it really doesn't change based on the antigen that it's binding. The B cell receptor, like the specific, like when you find a specific antigen, and again, we'll talk about this on Thursday, when you find a specific antigen, the FAB portion is going to like change to fit that antigen like perfectly. Like again, like a lock and key. And that's what's conferring specificity. That's like what is conferring like what this antibody can recognize. So and that's also, I just tried to give you all like the definitions, and that's what's specifically binding to the epitope on the antigen. Remember the epitope is like that amino acid. It's like an amino acid sequence. So again, it's like a lock and key. And then some other just like things I was trying to think of to like show you. So the hinge region is here, it's in the middle, and I tried to tie it into biochem. So there's a lot of proline amino acid within the hinge region, and this is kind of what allows it to be flexible. So it's allowed to kind of bend and be flexible because of all that proline there. There's also some disulfide bonds, and that's kind of what make up like the arms. Well, not the arms, but the FAB like portions of it. Yeah. And it's, I think, like, yeah, they did do this, but the variable region, so the variable region is what's like colored in orange, and you'll notice it's the same for the B cell, like the B cell receptor and the T cell receptor. The variable region's in orange, and then the constant region is in blue. And this is why I don't think FC stands for like fragment of like the constant. Don't get confused, because the constant region actually goes up into like the FAB portion, right? It goes up into, it extends up into it. So I don't want you to like get confused with that terminology. Okay. Versus a T cell receptor. So you can kind of think of a T cell receptor as like basically just being like one arm of the B cell receptor, which is what it kind of is. If I go back really quickly, it kind of looks like we just took this off and we're like, okay, here's your T cell receptor. Usually, like in T cells that we know, they're made up of alpha and beta chains. Some T cells have gamma and delta chains. We'll talk about that on Friday. There's like it's like a terminology concept called race for rearrangement. We'll talk about what that means on Friday. But the gamma delta T cells are like involved in like a certain developmental process. But these are big buts. Unlike B cell receptors. So B cell receptors, remember, those are like antibodies. They can get secreted. The B cell can secrete them if it needs to. T cell receptors are never, ever, ever, ever released. They are not released. They can't get secreted by T cells. They are stationed on the T cell. And another thing is, like, antibodies can recognize antigen on its own, right? Like, that's why, we're giving people like the antibodies right now with COVID, because it can find COVID, it can bind to it, and kind of, we'll talk about some functions of antibodies in a little bit, but it can find COVID, it can bind to it, and it can kind of like, it can recognize it on its own. T cell receptors they actually cannot recognize antigen on its own. It needs to be presented, must be presented antigen in the context of MHC. This is super important. I even highlighted it. This is going to come up like constantly throughout like understanding like T cells, T cell activation, autoimmunity. Like you need, like when you're presenting antigen to a T cell, if you just give it like, here's a piece of pollen, the T cell receptor is not, it's not even going to give it the time of day. It's like, cool, I don't know what this is. It needs to be presented in the context of MHC. And I'm going to show you, like we talked briefly about like MHC class 2, right? Like after it digest the thing, it's going to load it onto like an MHC. and we're going to talk about that, but that's kind of what I mean. It needs to be like loaded onto something and presented to the T cell. And it's just a way, like yesterday we talked about like the immune system really needs to be regulated because you don't want like active T cells going around and just going like killing all of your cells. So this is kind of a way, it's just like another fail-safe mechanism just to kind of really make sure like, hey, do you really want this T cell to be activated? So it's like, yes, I do. I've loaded it. I've taken the time to load it onto an MHC. Okay, so that's kind of why it's really important to understand this. Like, this T cell will not just recognize these antigens if it's not broken down and then loaded onto an MHC. And we're going to go into that. This slide's confusing. I'm going to go into it more on Friday. I actually have this exact same slide on Friday, but I did mention kind of like the T cell receptors in alpha beta, and then like there's also gamma delta ones. Oh, sorry. Disappeared. Do B cell receptors need MHC to communicate with T cells? Great question. Yes, they do. And we'll talk about that. But B cells, that's why B cells are antigen presenting cells. And we'll talk about antigen presenting cells. They have a specific type of MHC. And that's why B cells are considered antigen presenting cells, because they have this specific type of MHC. So it allows it to talk to the T cells. Yeah, good question. Okay, so this is kind of what I meant. This is kind of what I mean. So here's like your antibodies, right? So here are antibodies. They can just straight up, boom, paratope, epitope. So here's your paratope on the FAB portion. Well, I'm not pointing to it, but here's the epitope, the blue, that's the epitope. So the epitope is the part of the antigen that is basically recognized by a paratope. The paratope is on the FAB portion of the antibody. So if I'm looking at this, this is kind of flipped around. So here's the FC portion down here, and then here's an FAB portion. AB, and here's another FAB portion. And then like if I were to split this in half, this would be like the variable region. So the variable region is the one that can change and like really nicely fit to an antigen. So that's going to eventually make your paratope. And then the paratope is going to bind to the epitope on the antigen. So this whole thing is an antigen. This blue region is the epitope, and it's going to bind to the paratope of the antibody. But the epitopes recognized by T cells are buried. It doesn't like again, T cells need to see the antigens in the context of MHC. So this is going to get broken down, and then it's going to get like fragmented, and then the exact portion that the T cell needs to recognize is loaded onto an MHC molecule. And MHC molecules, we'll go into a little bit more detail, but they're just like on the surfaces of cells. And now the T cell receptor is like, okay, I got it. Like, you want me to actually mount a response against this. Okay, cool. So that's kind of what it's doing, is it's binding to both the peptide, the antigen, like the epitope peptide, the antigen basically, and then also the MHC. And I put this note down here because this is going to come up really important with like when we talk about like how you make sure you don't have like auto-reactive T cells. Note here that the T cell receptor, it's not just binding to the antigen, it's also binding to the MHC molecule. So this is something that's like checked for when you're developing T cell receptors. Again, this is going to be on Friday with T cell development and whatnot. But when you're developing the T cell receptor, you need to make sure it's like it can recognize an MHC molecule, because you can note here you have like little portions of it that are binding to the MHC molecule. Okay. And some people say like the T cell, like the T cell antigen, when it's loaded, it looks like a hot dog and a hot dog bun, and it 100% does. So this would be like looking down into the hot dog bun, right? So yeah, so that's how you can think of the T cell, like the antigens being presented to T cells. They're like hot dogs and hot dog buns. And the hot dog bun is the MHC molecule. Okay. So what's MHC? I just said this word like 1,000 times. So MHC stands for major histocompatibility complex. There's two types, type 1, type 2, class 1, class 2. Sometimes you just see MHC1 or MHC2. That's just what they are, the two types. They're encoded for by HLA genes. So it's kind of annoying, but sometimes people interchangeably use like HLA. So like instead of saying like MHC, they'll say like HLA. That's fine. They're the same thing. It's like the gene is coding for this MHC. I'm not sure why they didn't just call the protein this MHC complex. I don't know why they just didn't call that an HLA, but it's not a big deal. But you'll notice, like, we'll talk about this a little bit with like transplants, like transplant stuff. They do HLA typing before you do transplants, and that's what they're looking for. They're looking at these MHCs. So the whole purpose of what MHCs are is to sit on the cell surface and display peptides. And again, this is really important for vaccine development and B and T cell activation. MHCs don't present carbs and lipids. They cannot. It's only peptides. They cannot, they will not, they don't. Okay, so they're only presenting peptide fragments. There's obviously a different way to present carbs and lipid like antigens, but it's not. And that's why, too, all antigens are peptides because they need to be loaded into MHC. Okay, but we'll talk about like carbs and lipids a little bit. I think we'll talk about carbs and lipids with like B cell activation because B cells can like see them in certain ways. But again, antigens, they're peptides, and MHCs load only peptides. Okay, so MHC class 1 is located on every single cell in your body. So all nucleated cells. So nucleated cells are all cells in the body except red blood cells and platelets, but class 1 MHC is actually located on platelets. So people basically say all nucleated cells except red blood cells plus platelets. It's kind of confusing. So it's basically just all cells in the entire body except red blood cells. So that's MHC class 1. And that's why, hopefully this makes sense. Like this is why you would do like HLA typing before you get like an organ transplant, because you want to just kind of make sure that they're compatible to the new organ, because every cell in the body has MHC class 1 on its surface. Class 2 is only located on the 3 APCs. So do you guys remember the 3 APCs? Hopefully you're saying them in your head right now. But your macrophages, your dendritic cells, and your B cells. Oh my gosh, please put that. Did you put that in the big group? Yes, thank you. That's amazing. Yes, however, you do need to know these. That's beautiful, Vanessa. Yeah, so remember that. So don't get tricked, though. I'm going to say this now so you guys don't get got. B cells, macrophages, and dendritic cells, these guys are nucleated, right? So they're going to have both class 1 and class 2 on its surface. Don't get got. They love to be like, oh, like what's on the surface of this cell? It's two, like they have class 1 and class 2 on its surface. Because they're still nucleated cells. They still have nuclei. So they're going to have both types on its surface. It's not like the APCs only have class 2. No, the APCs have class 1, but they also have class 2. And they're the only ones that have class 2. Okay. Okay, so let's talk about MHC class 1. So MHC class 1 is the genes coding for MHC class 1 is HLA-A, HLA-B, and HLA-C. I put this in here because there's some like autoimmune diseases associated with certain HLAs, and there's like a chart in first aid. I think I bring it up when we talk about like autoimmunity a little bit later, but that's just what I wanted you to kind of just start to make that association. Like MHC class 1s are coded for like the HLA A genes or the HLA-B genes. There's one HLA gene, it's actually HLA-B27. It's associated with like five or six autoimmune diseases. That's what it's talking about. It's talking about this, apparently this B27 MHC class 1 is like implicated in a lot of these autoimmune diseases. So that's just that's why it's good to know like A, B, and C are the ones kind of associated with the MHC class 1s. Looking at the structure of it, and I tried to color code it again, there's one long chain, so this one really, really long chain, and then there's this one, so this one long chain, and then there's like one, like what they call a short chain. I think they look the same size, but it's fine. So there's a long chain and a short chain, and then there's an associated protein. It's called beta-2 microglobulin. It's not a part of the receptor, but it's like receptor. It's not a part of like the MHC class 1, but it's like it's always with it, if that makes sense. So it's not like attached. So I think this first aid picture is a pretty good one. It's just always going to be there. So sometimes if they want to even like test for MHC class 1 for whatever reason, usually just in board style questions to see if you know like beta-2 microglobulin is associated, they'll say, oh, there's tons of beta-2 microglobulin present or whatever. And you're like, okay, MHC class 1. Like it's like a way to kind of identify that they're talking about an MHC class 1. I'll see if I can find an example of a question to show you what I mean. So again, MHC class 1, all nucleated cells, including your APCs. Please don't get got, including APCs and platelets. And MHC class 1 is basically like how the body distinguishes self from non-self. So it's going to present antigens to CD8 positive T cells. So present endogenous antigens to CD8 positive T cells. Endogenous, another way to say this is cytosolic. So again, we have like the cells in the body, if they engulf an antigen, right? So it's like an antigen. Let's say the virus example. So now this macrophage ingested this virus. Oop. This would be like an endogenous antigen because it's an intracellular thing. And so when it like breaks it down and puts it on its surface, it's going to load it into an MHC class 1 molecule, and then it's going to present it to CD8 positive T cells.
So this kind of like makes sense because you said the CD8 ones are the cytotoxic
Mm-hmm.
ones. So
Yes,
like if it's inside the cell, there's nothing you can do to get it out. You just want to kill the whole cell.
exactly.
Okay, that
Yeah,
makes sense.
perfect. Oh, good question. Why doesn't the body attack red blood cells if they don't have MHC class 1? Very good question. We'll talk about that a little bit more tomorrow with complement, but there's other receptors that kind of tell like, hey, like I'm one of you guys, don't attack me. And so the red blood cells have some of those other receptors on its surface. So it doesn't get attacked. That's a good question. So they call this the rule of 8, and we'll see it plays along with the MHC class 2s as well. But the rule of 8 is that MHC class 1 presents to CD8 positive T cells. So 1 times 8 equals 8. Okay. So this is kind of what I mean here. Here's kind of like a loading. This is like how it happens. So here's an MHC. So again, this is like, again, think like your intracellular things, like your viruses. Viruses go on, but it's not always viruses. It can also be like some parasites, just intracellular pathogens. So it's going to come into the cell. This one's going to enter with endocytosis. It's okay if you don't know what that is. It's not a big deal. It just comes in. It just gets engulfed. It comes in, and it releases its antigen into it. So the cell itself is going to basically break down this protein that it just injected. So viral proteins are synthesized in the cytosol. The cell is going to basically break down these viral proteins and then load them onto MHC class 1 in the rough ER. You should know this location. And the exact like transporter that loads MHC class 1, that loads peptide onto MHC class 1 in the ER is called TAP. And TAP stands for transporter associated with antigen processing. I tried to highlight all the differences between MHC class 1 and 2, and this is like a big one, is like the whole endogenous aspect, where it's loaded, and what loads it. So MHC class 1, these are molecule, like antigens are going to be derived from the cytosol, so endogenously. They're loaded into the rough ER by TAP. And then as they kind of make their way up to the surface, they're less stable than MHC class 2. Like after they're made, they kind of have to go to the surface, right? Because they have to like be on the surface so like the T cells can find them, the CD8 positive T cells can find them. I remember that class 1 trafficking is less stable than class 2 because I think of MHC class 1 as only having like one little line, whereas class 2 has like two little lines. So it's like, oh, it's more stable. That's not why, but that's good enough. That's good enough. You don't have to go into too much more detail. You don't have to know too much more detail than that. But as it goes to the surface, it's less stable than class 2 trafficking. And I only mention that because some microbes know this and they actually try to inhibit this surface trafficking of MHC class 1. You'll actually see like some cancers and some microbes can downregulate MHC class 1. Because basically what MHC class 1 is doing is like, hey, look what's inside of me. T cell, come kill me. Like, come kill me. Look what's here. So obviously like the bacteria and the viruses would want to downregulate it. So it's like, oh, you can't find me. Sorry, T cell can't find me. Can't kill you. So that's why I mentioned the stability aspect of it. It's kind of like targeted a lot by viruses as a way to evade the immune system. Okay, so this is kind of it's a little blurry. This is I think from an Anki card. I really liked it. So this is kind of the whole what we just talked about. And here's like the three main differences between class 2 loading. So the viral protein, so an endogenous antigen is going to be in the cell. It's going to be cytoplasmic. It's going to get loaded in the rough ER by TAP. So TAP is going to load it in the rough ER, and then it's going to traffic to the surface. And then it's going to be put basically on the surface of this cell, and then a CD8 positive T cell is going to come over and recognize the antigen. MHC class 2 looks different. So there are two equal length chains. So there's two alpha chains here, and then two beta chains here. And then what's associated with this, it's not shown here, but it's called invariant chain. It's like the associated protein. So beta 2 microglobulin for MHC class 1 is associated with MHC class 1. Invariant chain is associated with MHC class 2. The genes coding for class 2 they have 2 letters now. So remember the class 1s, it was HLA-A, HLA-B, and HLA-C. Now for class 2, it's HLA-DP, HLA-DR, and HLA-DQ. And again, same concept. There's certain autoimmune diseases associated with like, HLA-DR35 or whatever, or DR3. And it's like, that's what they're talking about. So it's like, those are the ones associated with the MHC class 2s. So again, that's what I just kind of wanted to give you context as what they're talking about. And there's like these genes that are like associated with certain autoimmune diseases. Okay, so the 2 equal length chains, again, MHC class 2, 2 equal length chains. The associated protein is invariant chain. These are only on your APCs. And what they're doing is they're presenting exogenous antigens to CD4 positive T cells. So the rule of 8 still applies. 2 for MHC class 2 times 4 equals 8. So that's the whole idea of the rule of 8. And exogenous in this instance is just like not in the cytosol. So like specifically for MHC class 2 loading, it's done in the lysosome, but in that way, like exogenous versus endogenous is just not in the cytosol. So here's kind of basically the same idea as the last one to show you what happens. So some sort of exogenous antigen, like a bacteria is a good example. It's going to get engulfed. Again, remember the macrophage, or like one of those phagocytic cells is going to phagocytose the bacterium. It's going to bring it in. And then what basically happens from here is, remember, we get the phagosome, and then remember the lysosome kind of comes over and it can kind of fuse with it and start to break down that process. So this is called an acidified endosome, but it would also kind of be like it's becoming acidified from the lysosomes. And the reason it's called an endosome is because this process is endocytosis versus like phagocytosis. So this could also be an acidified phagosome if the cell engaged into phagocytosis and then was fused with lysosomes. Does that make sense? Did I explain that really hard? So if the cell is like phagocytosing the bacteria, so it's going through phagocytosis, let's say it's going through phagocytosis here, it's going to bring it into the cell, and then it's going to go through that same process. The lysosome's going to come over, fuse with it, you get your phagolysosome, that's going to acidify the environment of the phagosome, and then you're going to get destruction. If the pathogen's brought in with like endocytosis, which is what they're showing here, I think they're showing endocytosis. Oh no, this is endocytosis. They're showing endocytosis here. Regardless, it's like it's the same thing. It's just terminology. I just don't want you to be like, well, where did endosome come from? It's just like whatever. It's the same exact thing. It's just like this one can be like the acidified phagosome, one's the acidified endosome, depending on which way it came in. But still, the process is still the same, regardless of which way it came in. So something is getting acidified from like the lysosomes coming and fusing. Something is getting acidified. And at that point of acidification, it's loaded like right there. So it's loaded exactly where like when the lysosome drops the pH and starts to make it like really low pH and acidifying the environment to try and destroy like the pathogen, that's when you can get MHC class 2 loading. And this is done by removing invariant chain. So remember I said invariant chain is like what's associated with MHC class 2. It's removed and then it can basically get loaded by something called CLIP. Okay. And this is more stable than class 1 surface trafficking. Again, it's the same process, but now it's just where it's happening is different. The protein that's loading it is different. And it can be phagocytosis, it can be endocytosis. It's the same process still. It's getting loaded within that endosome, lysosome, phagosome, whatever. It's getting loaded in that, not in the rough ER. So here's I just think this is a better picture of it. So this is kind of showing, so this is showing an endosomal pathway, right? So here's an exogenous antigen coming in. Now it's in an endosome. It's going to fuse with the lysosome. And then when that happens, when that pH drops, we're going to get loading onto the MHC class 2 molecule. So, and that happens by getting rid of that invariant chain. It's not showing CLIP on here, but once you kind of remove invariant chain, CLIP can kind of load this endosomal like pathogen onto the MHC class 2, and then that can traffic to the surface. Okay. Okay. Oh, I don't think I mentioned what this B is. If you see this B, this is my buzzword B. So whenever I put a buzzword B on a slide, it's like a buzzword for a certain thing. So meaning like if you see these terms, like when talking about like MHC loading, they're talking about class 2 loading. So that's just my buzzword B. It's like they'll always mention these things with this particular topic we're talking about. And then this is kind of what I was talking about. I did end up memorizing these for my exam, the like Immuno exam. I don't remember if that was important. I would definitely say B27 is huge. B27 is huge in terms of like being associated with certain things. I would just for safety know these. I was doing the Anki deck and they ask you these all the time in the Anki deck, but it's sometimes they'll just straight up ask like, oh, which HLA is associated with, I don't know, lupus. So it's like DR2 and DR3 is associated with lupus, right? And they give you like little ways to remember it, like 2, 3, SLE, right? So it's kind of good to know these. It is annoying, I know, but like just try to make flashcards. Just try to make flashcards to help with that. But this is what I was referencing with like certain HLAs are associated with certain disease. They can't say they're like they cause the disease, but it's like they're more likely than not. If you have an HLA B27 mutation, you're gonna have one of these diseases. I missed a question. MHCs are always initially located intracellularly. Yes, so MHCs are proteins, right? So they're proteins, so they're always going to be coming from the rough ER. So here's actually class 2, made in the rough ER. It's a protein, so it's being made in the rough ER. Then it's getting sent to like vesicular trafficking through the Golgi, right? And then it's going to make its way to an endosome. Same with class 1. Class 1, it's a protein. It's made in the rough ER, and it just so happens like as it's going along the way to go up to the surface, it's like TAP is just throwing antigen onto it as it's trafficking to the surface. But yeah, so since they're proteins, they're just always going to be made in the rough ER. And they're coded from like the HLA genes, right? So it's like, transcription, translation, all the fun stuff. But okay. So then we have our NK cells. So I said we were going to come back to NK cells. So let's say that a virus thinks it's being smart and it tries to stop MHC class 1 trafficking as it goes to the surface. So it's downregulating MHC class 1. And it downregulates it. It's like, great. Now a T cell can't find it. Like a CD8 positive T cell can't find it. It can't kill me. It can't do anything. NK cells are actually on the scene. So they're innate cells. They are innate cells. And basically what they're doing, and I'll show you a picture, but basically what they're doing is they're looking for cells that don't have MHC class 1 in its surface. Because again, like I said, all cells have MHC class 1 on its surface. So let's say here's like a regular cell. Let's say a virus tries to downregulate MHC class 1. So now it's not even there. So the virus is like safe in here and it's hiding. The NK cell is going to come over to it and be like, wait, where's your MHC class 1? And it's going to get activated to kill this cell. I know, it's amazing that we have any diseases, right? It's just crazy. Like the immune system thought of everything. But yeah, it is an innate immune cell. This is why I say that NK cells are like the T cells. They're like the CD8 positive T cells of the innate immune system. And hopefully after we talk about T cells on Friday, you'll be like, oh yeah, like they are. But I really think of them as like the CD8. They do exactly what CD8 positive T cells do, but just in the innate immune system. They even secrete the exact same enzymes that CD8 positive T cells secrete in order to kill the cells. Like they're the exact same cells. But remember, I talked about yesterday, like lineage-wise, NK cells derive from lymphoblasts, right? Remember I talked about how like the lymphoblast breaks off into B or T cells. They also break off into NK cells. So it makes sense that they're pretty similar to T cells because they come from the same cell, the cell line. So another thing I kind of talked about like this, when are these cells activated? Absence of MHC class 1 on its surface. And instead of like having this downregulate, so no MHC class 1, it has to also be induced to be active. So again, this is kind of like back to that red blood cell question, right? Red blood cells don't have MHC class 1 on its surface. It has other things on its surface to tell the body like, hey, don't kill me. I'm cool. Like I'm you. One of those things is it doesn't have like this activation signal for NK cells. And I'll kind of show you what I mean by that. So that's like one way you can activate NK cells. You either like downregulate MHC class 1 or you have just like an activation signal on the surface of the cell. A lot of the times these activation signals are like stress signals, and it's like like the cell's like last way to try to be like, hey, like something's going on in here, and they'll try to like upregulate certain things on the surface. Another thing that they do is called antibody dependent cell mediated cytotoxicity. That is a mouthful. Antibody dependent cell mediated cytotoxicity, but it tells you exactly what the process is. So ADCC, it's basically there's an antibody involved. So an antibody is going to bind to a certain cell, and then it's going to trigger cytotoxicity, so like destruction of that cell. Eosinophils also use ADCC, and I'll show you like what that is in like in a few slides. So here's what I mean. So here's an NK cell. It's going to have an activating receptor, which is going to activate the NK cell, and it has an inhibitory receptor, which is going to inhibit it. So here is a normal cell. MHC class 1 is going to bind to the inhibitory receptor, and the NK cell is not going to get activated because it's like, okay, this cell has MHC class 1 on its surface. This must be like a normal cell. I'm not going to kill it. Because basically, remember, it's looking for cells that like have downregulated MHC class 1 to try and hide from like T cells. It's kind of like, no, like no, it's not happening. So in the absence of this MHC class 1 molecule, now there's only an activating ligand. Now we get activation and we get killing. Okay. So that's kind of like what like cancer cells try to do, or virus cells. They try to downregulate MHC class 1. NK cells kind of swoop in and kind of like kill them because of that. So with ADCC, there's a particular antibody not antibody. There's a particular receptor on the NK cell. It's called CD16. I don't know if this is like a need to know. I feel like this is like a very specific, question. So if a cell is missing the activating ligand, will it be invisible to NK cells? Yes. And that's why NK cells don't always catch like certain, like that's why we have cancer, right? Like it can still evade NK cells if it completely gets rid of these activating ligands too. It'll just completely be invisible. And that's actually why cancer cells like grow in like perfect harmony up until like a certain point. And that's when they're like, oh, you have like cancer, you've had it for a while. And you're like, what? Like I had no, you know what I mean? So that's one way that the cell can do that. It's a good question. Yeah. But a lot of the times like viruses especially aren't that smart. They just downregulate MHC. So they're still like seen by NK cells. Okay, so CD16, I would just go ahead and know it. It's like you don't have to know enough stuff, right? But the reason why this is an important thing to know is because this is the receptor on the NK cells that's involved with this ADCC process, the antibody dependent cellular cytotoxicity. So what happens with ADCC is here's a microbe and it's like coated with antigens. And let's say there's a IgG antibody that's going to bind to it. And I know we didn't really we're talking about antibodies on Thursday with like B cells, but IgG is going to just coat the surface of this antibody, of this pathogen, okay? CD16 on the NK cell recognizes the FC portion of the IgG antibody, okay? So if we draw an IgG antibody, here's FAB, right? There's two FABs, FAB. Here's FC down here. So NK cell CD16, so here's CD16, here's my NK, CD16, it's going to recognize this bottom portion of the IgG antibody. And that's why a lot of the times you'll see like, oh, CD16 is an FC gamma receptor. And what that means is FC of the IgG or the Ig gamma. Like that's what G stands for, is gamma. So that's basically the same, like this is the same term, FC gamma or IgG FC receptor. It's the same term. The gamma is like the G in IgG. If this was an IgA antibody, it would be FC alpha, right? So the FC gamma portion is the FC portion of the IgG antibody. So now when this pathogen is coated remember the FAB portion is the portion that binds to pathogens. That's what the portion that recognizes antigen. So now, the NK cell comes in, it binds to the FC portion with its CD16 receptor, like multiple NK cells can come in and do this. It binds to the FC gamma receptor, and now it's basically like enhancing NK cell killing. So it's almost like the NK cell was told, like, hey, I got one, right? I got one. Can you come kill it? So it's kind of like now it comes over and it releases perforin and granzyme. This is the same exact thing that CD8 positive T cells release to kill their targets, and then the cell will die. So that's what ADCC, antibody dependent, we have an antibody, cellular cytotoxicity. We have a cell and we have a dead cell. It's cytotoxic, right? So that's kind of what ADCC is. NK cells are famous for engaging in ADCC. Eosinophils can also engage in ADCC. So eosinophils, again, we'll talk about this on Thursday. Eosinophils have respond to IgE, and IgE is really big in like parasitic infections, allergy infections, everything that like eosinophils are like also involved in. So here's like here's an IgE. Same thing, has FAB portions, right? So FAB, FC portion down here. Now this would be FC epsilon. So that's kind of what this is saying. So eosinophils have a receptor that I guess you don't need to know the name of, binds to the FC portion of the IgE. So or the FC epsilon. So it'll come in and it'll recognize this and it'll bind to it. And then it can also engage in antibody-dependent cellular cytotoxicity. So basically, antibody-dependent cellular cytotoxicity, ADCC, is anytime you're having like an antibody binding to a target and it's enhancing the killing ability. Like just the fact that this is here is enhancing the killing activity of like a certain other cell. That's like what ADCC is. Okay, and eosinophils too, we'll talk about on Thursday, but they're really, really big, and IgE are really, really important for getting rid of helminths, or which is just like worms, various types of worms in the body. And you can also see here like multiple cells combined to the same antigen. Like you can have multiple antibodies. Like you can have like 10 eosinophils if we want, right? So it's not like yeah, you got the gist. Okay, so summary before we do some questions. There's two integrated systems, the innate and adaptive immune systems. Important innate cells are going to be your dendritic cells. They're phagocytes, but also antigen-presenting cells. Most important, only one that can activate your naive T cells. Macrophages are also phagocytes and APCs. Your PMN, I think I forgot to mention this, I'm sorry. PMN is another name for a neutrophil. So it stands for polymorphonuclear cell. Super annoying. But neutrophils, like they have like multiple lobes of their nuclei. So they have multiple lobes. They have like 3 to 5 lobes. So they're saying polymorphonuclear, right? So there's just many like shapes of their nucleus, which makes sense because there's many lobes. So if you see PMN, I'm referring to neutrophils that kind of do that like back and forth. Those are your, so PMMs are really, really good at phagocytosing. They're like the best ones. And then the mediator releasing cells, your basophils, are in the blood. Mast cells are located subcutaneously. They're in the tissue. And then eosinophils are both blood and subcutaneously. They're weakly phagocytic, but primarily they're releasing those mediator, like these are mediators, right, that it's releasing to try and kill the helminth. And if you have an increase in eosinophils, I'm sure you guys might have maybe done an LI on this, like maybe at some point in the past. Increase in eosinophils, you want to think parasitic infection. It's like a classic. It's not always, but it's like a classic indication. And then very briefly, because we're going to go into a lot more detail, important adaptive immune cells are your T cells, main two types, CD4, CD8. CD4 binds to your MHC class 2s, and then CD8 binds to your MHC class 1s. And then your B cells, remember, B cells can also be antigen-presenting cells. When they get activated, they turn into either both memory cells and plasma cells, and we'll talk way, way more about this on Thursday. Okay. Do you guys have any questions before we do some like questions to kind of show you what you should have taken away from today? Okay, well, okay. Okay, so we're gonna start with a review from yesterday. See how much you guys remember. Same thing as last time, just send them. We can just send them in the, you can send them in the chat. And it's more so to just kind of get an idea of when you guys are starting to slow down. So I don't slow down before anyone is done.
Oh, I think Dawn might have asked in the chat if we could get a 5-minute break before the questions.
Oh yeah. Oh my gosh, yeah, sorry.
Oh, oh,
Absolutely.
oh no. So I actually meant like not for today. I guess going forward at like 7 o'clock after an hour, if we could just get like 5 minute break.
Oh
We don't
yeah.
have to do one now. I'm just saying like for future lectures, if it's cool with everyone else, obviously,
Yeah,
and with
I
you.
don't mind. I don't mind at all, yeah. As long as you guys don't mind like going maybe a little over with the questions. I mean, 5 minutes isn't gonna do much, but yeah, I don't have a problem. It'll help me. I always have to go to the bathroom. Always have to. All right, so you guys are slowing down a little bit. Okay, so you guys are like exactly 50-50 split on this. It's so funny. Sorry, this is my really bad attempt at writing a question. So this is actually going to be a B cell. This area it's pointing to is your follicle slash the germinal center in the middle of it. Remember? So remember the follicles are like the circles, and then inside, if it's activated, it has a germinal center, or you can call it a secondary follicle. That's kind of what it's referring to here. I wanted to put this picture on here because I think people like when you think cortex and like, it's like cortex for the B cells and like the paracortex for the T cells, right? I think people think the cortex, it has to be all the way on the outside, which is not always the case, which you can see here. So I just kind of wanted to like emphasize that. So the paracortex in this example would be anything that's not like where a follicle is. So it would just be the portions in between. And then like something like this would be like the sinus. Like remember we talked about like the sinuses and the cords. That's what like these guys would be. Okay, so when you see like these follicles spread out throughout a lymph node, these are all follicles, these are all areas of B, like where B cells are. And then the ones that look like they have like a little secondary circle within it, that's your germinal center. That's gonna be like your activated B cells. Oh, is the ring around the follicle T cells? No, good question. So the ring around, so the follicle, then we have like the germinal center in the middle, right? The paracortex is going to be, it's not like the ring, it's just going to be the areas between the follicles. So that's why it's not like cortex medulla. Like that's not why like T cells are located in the medulla. It's just like around the cortex. So paracortex, it's just like around the cortex. It's like not a cortex in the way you think of like traditional cortex medulla, like from like anatomy, from like just anatomy throughout, med school so far. It's just a weird thing with lymph nodes. The cortex is just where where the follicles are. The actual like circle, the actual ring, this would be like the marginal zone, and that's where the B cells and the T cells meet, but that's because the T cells are like out here, right? And then the B cells are in here. So that's kind of where they meet. They meet at like this marginal zone. Yeah, no problem. Yeah, that was like something I just kind of wanted to re-emphasize because it's like a weird thing with lymph node. It's like a weird thing with secondary lymphoid tissue. And I feel like every image you see of it, like the follicles are still up like where a cortex would be. Like they're always like out here in like a perfect array, and it's not always like that as you can see here. So that's kind of why, yeah, again, I wanted to show you. And this is like an actual biopsy too. So I wrote here again, I just kind of made this up randomly. So this is kind of like a germinal center of a follicle, which would be the B cell zone, aka like the cortex. And this is located in the lymph node cortex. Yep. Okay. Next. Okay, I'm gonna go through this one, just slowing down a little bit. Last chance. Okay, so this one is if you just read the last sentence, basically, which of the following receptor types is responsible for initiating the inflammatory response? Inflammatory response, innate immune system, right? Thinking like, okay, we know we will get like an inflammatory response. We'll get like that innate immune activation when we get our PRRs binding to our PAMPs, our pattern recognition receptors binding to the pathogen associated molecular patterns. That's one of the things that happens in the innate immune response. So one of those examples of a PRR is a toll-like receptor. Another answer that could have been here, which also would have been right, is like a NOD receptor or an NOD. Well, it's the same thing, but like NOD receptors are also examples of PRRs. Okay, and I put this here. It's kind of the same thing. And once these are uploaded, you can go through all of these again. But yeah, PAMs bind to PRRs that initiates like the innate immune response, including the inflammatory response. Okay. Okay. I think a majority of you guys have answered. How long do you guys usually give each other when you're doing questions, like during case or during LIs? Or does it not? Oh, so like it, okay. Okay. All right. So we'll just get, yeah, exactly. Yeah. So I think that's what I'm going to do too then. That's what a lot of you guys are saying. So you guys get 90 seconds on the exam. I'll give you guys 90 seconds. But also, I know you guys have just like learned this for like the first time ever, but I do just want to try and get through a lot of questions just so you can kind of see how things are asked. Because a lot of the times they're not just going to be like, what is a toll-like receptor, right? So I just kind of want to show you how questions are asked. But I'll try to stick to the 90 seconds, even though I know that's probably not enough time for just learning it for the first time. Well, for some of you. Okay, so for this one, you guys are split a little all over the place, which is good. That's kind of the point of these. So this is mentioning there's a dysfunction in the NOD2 receptor. NOD2 receptor, this is a PRR. Remember, I just kind of like threw that in there. Like a PRR can be a toll-like receptor or a NOD2 receptor. So when PRRs bind to PAMPs, remember I said you get activation of the inflammatory response. That's like one of the things that can happen. And that's kind of what it's saying. This is involved in an innate immune system response to PAMPs. So for this one, it's actually going to be NF-kappa B. Of course, I threw an NF-kappa B question on here, right? Yeah, so remember, NF-kappa B is one of those things like that has so many functions in the cells, but one of the things that it does is as soon as you get like binding of some sort of PAMP to a PRR. So remember my little like animation for like LPS binding to a TLR. It's the same thing here. Like NOD2 is also a PRR. You would also usually get NF-kappa B activation. Okay. That's kind of
Can
what.
I?
Yeah,
Oh, sorry. Okay. I'm just trying to keep everything straight in my head.
I know. There's a lot.
So I wanted to clarify where are these nod receptors? Like what type of cell are they sitting on?
So they're also still on any immune cell. A lot of the time, they're like the sentinel cells, like your macrophages, epithelial cells, like those sentinel cells that are like sensing infection. But a lot of cells have PRRs. A lot of cells are kind of capable of doing this. Um, yeah,
And
so
then
I would stick to like the sentinel cells.
the PAMPs that they are recognizing, I mean, that's just a general term for just something that's abnormal, right? That's
Right.
not a specific structure that they're binding to.
Right. A PAMP is it's just like, that's like a fancy way of saying like something that bacteria and fungi and viruses have that we don't have. So even in like this Amboss explanation, they say LPS, flagellin, peptidoglycan, double-stranded RNA that belongs to viruses would be one. It's just anything that's like we don't have in our body can technically be recognized by a PRR and is technically considered a PAMP.
That's really helpful. Thank you so much.
You're welcome. I know there's a lot to keep track of. I'm very aware. That's why I tried to break this down like this and not just scare you guys with what we're going to do on Thursday and Friday first, because you're going to be like, what is happening? So just kind of wanted to give you a little bit of an intro. But yeah, so nod receptors and toll-like receptors pretty much do the exact same thing. It's just their location is different, but it doesn't matter. It's like you can think of them as like being the same thing. So if this had mentioned a I don't know, like TLR4, this had said, okay, TLR4 is deficient, it would still be the same answer, if that makes sense.
And then I'm sorry, just to clarify one more thing. Then PRR is just a general term for nod to,
Yes.
and then the toll-like receptor.
Yeah, it would be like TLR. So example of pattern recognition receptors, TLR, and nod would be on here, like nod receptors. Immuno, they just like to make names for everything. So they like to give things names. And if it was up to me, I wouldn't say any of these names, but this is how they ask this is how they ask questions. So it's good to at least like be aware of them. But kind of the same thing with PAMPs. PAMPs is like a category of just it's like an all-encompassing list. It's anything that's like in a bacteria or a virus or something that we don't have. And then these guys are what's recognizing the PAMPs.
Thank you.
No problem. Yeah, I'm glad you're asking questions because I'm telling you, everyone's probably like the same, thinking the same thing. Immuno's hard to get at first. There's just a lot of terms, you know? Okay. Good with this? PRRs is toll-like receptors, nods. Okay. Next one. All right, you guys are slowing down. Love when you guys like switch your answers like 3 times. Okay, so this is a little tricky. This is a little like tricky way of them asking it, but again, they're not just gonna be like, what is the function of IL-2? Like they're just not gonna do that. But that's all this is asking, basically. It's like, okay, you're giving IL-2. Why would you give IL-2? Hot T-bone steak. This is T in hot T-bone steak. We're stimulating T cells. That's all it's doing. These are all these other guys are just fancy things. They're very good distractors. So I like these boards. These are from Boards and Beyond. These Boards and Beyond questions have really good distractors. But all you're doing is, yeah, like we didn't say it's not specifically CD8 T cell stimulation, but it's T cell stimulation, and like yeah, by stimulating T cells, you're stimulating CD8, right? Like you're gonna eventually do that. So that's all this is kind of asking is like the function of IL-2 is your T-bone like the T in T-bone steak, and that's like your T cell like growth factor, right? So. I think that's actually like another name of it. Yeah, IL-2 is famous for its role as a general T cell activator. So again, it's just like a whole, it just activates T cells. That was a tricky one though. That was a tricky one. Couple more. Okay, so that was like 90-ish seconds. So, um, again, I know that this is really quick. I get that. You're gonna be able to do all of these again, I promise. But for this one, so it's saying those endothelial cells of the blood vessels have increased numbers of cell adhesion molecules, those CAMs. And yes, this is like a nitpicky thing, but it is one of the functions of IL-1. Yeah, some of you guys, you're like, wait. Yeah. So this was a tricky one though, because she's got a bunch of stuff going on here. You would think like, okay, swelling, like that's gonna be like your IL-6 maybe, or your stuff like that. But when they're talking like specifically about your cell adhesion molecules, that's gonna be IL-1. And I know it's not in hot T-bone steak. That's what I was kind of meant. I think that's why I threw this one in here, just to be like, hey, just don't forget, like there's other stuff that like these cytokines can do. But that's kind of what he's saying here. Some of you guys picked IL-8. Which is good. I mean, that's also going to happen. But I think because they were just kind of mentioning the upregulation of the CAMs, that's going to be your IL-1. And then just right on the acute then recruit, it seems like so long ago, activates the endothelium. So it activates these CAMs on the surface. Okay. Okay, let's go over this one. You guys, when you have like combos in the text box, it's like exactly what I do. I'm like, but wouldn't it be this? Okay, so this one, you didn't have to read the whole thing. Which of the following is important for clearance of viruses? Viruses, endogenous, right? These are endogenous antigens, basically. Endogenous, I just really want you guys to start associating viruses with your MHC class 1s for clearance. Your CD8 positive T cells are going to come in, take it out. If MHC class 1's not there, NK cells can take it out. A lot of you guys did say macrophages too, which is not wrong. I think it's one of those things that's annoying, but it's like, oh, like D's a better answer, right? Macrophages, they are antigen presenting cells, so they can technically be infected by a virus, but they're still going to load it onto an MHC class 1. Sorry, they're going to load it onto an MHC class 2, sure. But I think like in general, that would be the most important. Is like you want to associate class 1 with your endogenous antigens like viruses, which is kind of what he says here. He's like, yeah, like macrophages do play an important role, but viruses are kind of like, unless the virus is infecting the macrophage, it's kind of like it's hidden because it needs a cell to be able to replicate. So, and this is kind of what we had talked about. Make sense? Okay. 2 more, I think. Sorry, we're a little over. Okay, guys ready? All right, let's go over this one. So this is basically saying this virus gets rid of MHC class 1. So if you get rid of MHC class 1, what cell would be inhibited? What cell would not work? And if you get rid of MHC class 1, you get rid of your CD8 cells, your CD8 positive T cells. Remember the rule of 8. So MHC class 1 binds to it. A lot of you guys pick CD16. Technically, I think you just read it wrong, because you'd be right if it was saying, like, if MHC class 1 wasn't there, what would be activated, right? You'd activate your NK cells. But a trick, and we didn't go over this, but if you just happen to know, CD56 is also found on NK cells. So you could have kind of been like, nah, like we could have kind of like ruled this out. But that's kind of what it's asking. Like you inhibit MHC class 1, so what cell won't work? And it's your CD8 positive T cells. Okay. Okay. And again, I'll send these out to you guys and you can go through them. Well, I won't, but Sarah will post them hopefully soon. Okay, last question. Okay. Last one. How'd we do? So remember we talked about, so this person has an abscess, right? So there's like a thick white liquid that's expressed, that's pus. So remember what makes up pus? I kind of briefly mentioned it. I knew that this question was in here. So I was like, oh yeah. Yeah, so pus, remember it's dead neutrophils. So dead neutrophils primarily make up pus. So remember neutrophils clean up on IL-8, right? So that's kind of what this question was getting at. So IL-8 is the main chemotactic factor for, this is granulocytes, but it's usually your neutrophils. And again, it's your cleanup on IL-8. One of you guys had asked about IL-14. Don't worry about IL-14. It's like, it's involved with B cells, but it's like not one of the ones that you have to know. We're going to talk about IL-2, IL-5, and IL-5 later in the week. Don't worry about these other two. That's why I didn't even put like, all of the answers on here. You don't really have to be, there's a lot you have to memorize. So I don't want to give you more than what you needed. Okay. All right. That was all the questions. So let me stop.